4:30.
Okay, we start recording. Go ahead.
Okay, got it. Okay, so I asked to start. I want to present to you few things that you bear in mind. Oded will give the major lecture today about how plants get the iron. But why it's important that plant will get the iron? The major thing is that iron in the food, in the plants, is a very important source for the iron that we get in our body. Because the iron that we get from meat, from tissue, that non-vegetarian or non-vegan can get, is mainly through hemoglobin or through myoglobin, different sources. But there are many people that like to have a diet that is more non-heme iron. Cereals, fruits, legumes, vegetables. This is very important to basically also to people that like meat, but it is very important. Now, you will see again and again this picture in Yoav's lecture. He already mentioned it, that iron absorption takes place in the duodenum. It's the part of our colon, a very short part immediately after the stomach. And the reason that I put it here is to show you that actually the iron, the heme iron, gets into the cells of the duodenum a little bit in a different way than the non-heme iron. The heme iron gets through a heme transporter that gets it into the cells, and there it's kind of, most of it is processed, and the iron gets into the bloodstream. But the non-heme iron, the one from the fruit, gets through a cytochrome B or another transporter called DMT1. And you will hear about this a lot from Yoav. And the non-heme iron, the one that comes from plants, can either go the same way of the released iron from the heme, or it can go basically, it's lost in a ferritin, in a special ferritin that catch it. And from here, if it goes into the bloodstream, it's practically like the iron that comes from meat. So as I said, this you will hear again and again. But what I want you to keep in mind during Oded lecture is the fact that although plants are considered a full organism, like a cow, like us, it cannot move. So if the iron in the plant is so important, different mechanisms have to be developed. And we always think on plants that plants have everything they need because they do photosynthesis and they create their starch and they get the CO2 from the air. But there are 16 essential nutrients that plant has to require. And this, I will not talk about all of them. These are considered primary or secondary. I'm talking about the trace elements, something that the plants don't need in massive amounts, but here is the iron. The iron is something that the plant cannot produce themselves. They need to get it from outside. And this is exactly what you will hear today because it's so essential, because plants, they don't have iron. They turn very yellow. This is one of the ways that we see that the plant is in trouble. But there are a lot of mechanisms that Oded, who is our guest from Barcelona, and he's an expert not only in the plants, but also in the soils where the plants get the iron from. So Oded, the floor is yours. I'm stop sharing, and you can put your slides on.
Very much.
Yeah.
I'm very happy.
Then go please to the display mode.
Yeah. Can it be seen?
Yeah.
Okay. So I'm very happy to be here, to be able to give my part, share a little bit of my knowledge. And I'll tell you just in one slide who I am. I studied also in the Hebrew University of the Faculty of Agriculture in Chovot. Have passed my BSc in horticulture. If anybody doesn't know what it means, I'll say I wrote it down. That's the branch of plant agriculture dealing with intensively grown crops like fruits, vegetables, ornamental plants, nutraceutical plants. Does anybody know or can give an example what's a nutraceutical plant? Please jump. No.
Any plant which is being used in medicine. Does anyone know any plant?
Okay. Okay.
Of course you know.
Plant that is used in medicine?
Yeah.
Artemisinin, for example.
Very
Do you
good.
know it?
There is one that's now bursting all over the world and being legitimized all over the world as well.
Cannabis.
Exactly.
Cannabis.
Thank you. Great. So my MSc had to do with plant protection, internal and applied strategies for controlling attacks incurred by macro and micro organisms. And my PhD dealt with plant physiology and biochemistry. My main theme on my thesis was the production of ethylene in both higher plants and fungi. And I'm showing both categories of agriculture that I studied, just in order to encourage some of you who are still in the situation of students. So I think that it's very good to look at different items, different subjects from different angles, different points of view. So horticulture is one thing, which does not coincide with plant protection, which does not necessarily coincide with plant physiology or biochemistry. So if you study all, try to diversify your studies in order to get a more balanced look at your subject or in the main subject. Then I passed to work in the industry. I did for several years R&D in plant micropropagation. Maybe the Israeli participants know about Rahan meristem, Rosh Hanikra meristems, where we grew such plants just in agar and in other ways as well. Then I passed to work in another part of the industry. I managed the agronomical know-how of Haifa Chemicals, which produces specialty fertilizers. So I've got quite a good background regarding agriculture, but following Yoav's request, I've studied the current subject of iron just recently in order to produce this talk. So just bear with me if not always I'll be so fluent, and if I hope I won't make any mistakes, but such things happen, and don't worry, you can correct me the moment you feel that I have done any mistake. So where are we? Of course, iron starts being absorbed by a plant from the soil. So iron is the second most abundant metal in the Earth's crust after aluminum. And just like Rachel just mentioned, there are 16 nutrients, but aluminum, obligatory elements, but aluminum is not one of them. So basically, iron serves as the most ubiquitous plant nutrient on Earth. And you can see here, I hope, okay. So when you can see here very clearly, it's obstructing my view. Okay, I will remove. Okay, so it's very easy to see that the ferrum or iron is very common in igneous minerals as well as sedimentary ones, far higher than any other element. Why doesn't it move?
Page down. No, just page down.
Not either now.
No.
Okay. So yeah, passed a lot of them.
יואב, אנחנו רואים את המצגת כאילו לא בכל המסך, כאילו רק חצי מה...
אני יודע, אני אמרתי לו, עודד, אתה צריך לעשות את זה על מצגת, אתה עושה את זה עכשיו...
כן, אני רק לא הצלחתי להעביר את הסלייד.
אבל תלך למטה, או תעשה F5.
אוקיי.
אבל תלך למוד של מצגת, כי אחרת זה יוצא קטן.
ברור.
בדיוק.
אוקיי. עכשיו זה בסדר.
נראה לי שאתה צריך ללחוץ קונטרול L.
אני חושב שעכשיו זה בסדר.
לא, עדיין זה לא בסדר.
לא, זה לא בסדר.
קונטרול L יפתור את הבעיה, אני חושבת.
נכון.
How is it now?
לא. Control L.
This is what I did.
אולי כשהוא יוצא מהמצב של הצגה, צריך לעשות את זה?
אתה יכול לעשות הגדלת תצוגה.
כן, את צודקת, הוא באמת צריך לעשות סטופ שייר, אחר כך להתחיל שייר, ואז קונטרול L.
אוקיי, עכשיו עוד פעם, share screen?
share screen.
Control L. איך עכשיו?
לא רואים כלום.
אני צריך לעשות...
share screen.
או שאתה תראה את המצגת, והוא יגיד לך תעבור. זה יהיה הכי פשוט, כי הוא גם הקונקשן וגם זה מצגת עמוסה.
אני לא מצליח. לוחץ על share screen. אוקיי.
כן. בסדר, עכשיו זה טוב.
כן?
כן.
אוקיי. זה לא קטן לכם?
לא, זה קטן, כן.
אז אני אעשה שוב, אני אבוא עוד פעם
כן, וזה, כן.
פשוט שבאופציות של המצגת שלך זה presenter mode, אתה צריך להוריד את זה מ-presenter mode.
בדיוק.
תהיה מצגת רגילה, פשוט תלך לאופציות של המצגת למטה.
טוב, בואו נמשיך כרגע, אם אתם רואים עכשיו זה בסדר מבחינתי. אתם רואים?
רואים?
זה בסדר, אפשר להתמודד עם זה, זה לא
אפשר
כזה נורא.
להתמודד עם זה, כן. אבל תלך לסלייד המתאים פה, כי אתה לא נמצא בסלייד הנכון.
טוב, עכשיו זה שוב לא מגיב לי.
זה, אוקיי. אוקיי, now.
טוב, אז something we have talked already, Yoav has mentioned it very many times, so I'll just come up with reading it. Probably more than any other element, iron markedly influences the chemical and physical properties of soils and sediments in the earth. Considering its transition method status with potential variation in electronic configuration, ionic radius, and magnetic moment, combined with its abundance and relatively large mass, little wonder that one sees its unique influence on every hand. And this was said already 1988. So here we can have a look at what it means. How does we get in touch with iron in the soil? So we start with the volcanic ashes and volcanic lava and magma and all these situations. So it becomes weathering for people who are not familiar with weathering. In Hebrew, it's blaya or itrochechut. Does it take a belt for you? I can't hear. So everybody is now familiar with weathering?
I'm not sure.
Okay. So again, weathering in English, in Hebrew is blaya or itrochechut, meaning breaking big masses into small particles. And this takes place by various factors that I'm going to look into later on in the next slide. But here you can see that the bigger particles are found in sand. We are now talking about the structure of soil. So we have got different sizes of particles. Sand is meant by the biggest one. It's between 2 and 0.05 millimeters in size. Then silt, 0.5 to 0.002, and clay is the smallest particle. And together with this, we get a more intensive oxidizing of the ferrum. And also we can see various types of the minerals. Here we are looking at the weathering of soil minerals by aeration, water, and here microorganism for me is the center of this slide. We can see that in the lower part, the fresh zone where it's still untouched, and the upper part of this figure, we see the elemental compound, the leaching, the atmosphere, and on the middle areas we can see how microorganisms work. We have the green on the left, photoautotrophs that tackle carbonate ions and increase the alkalinity. Therefore, it's active in a chemical way. All withering organisms, including chemoheterotrophs, are here in the middle. These are in violet, and they excrete siderophores that we have heard some time ago in many of the previous talks. And they excrete between other things, they excrete organic acids, and thereby they increase the acidity, and also they serve as chelating agents. Also, they excrete catabolizing enzymes which solubilize the elements and compounds. And on the right-hand side, we have autolytrophs that biocatalyze and facilitate redox reactions. So it's a kind of a combined reactions, chemical and physical, and which produces salts which solubilize the soils, the soil, and with this it's withering more and more, and by this they form the soil. So here's iron in the soil, and what's the fate of soluble cations? Now I'm looking at cations because iron is a cation. So we can see that what's the fate of the different cations can be looked in various directions. For example, up top, we have got plant uptake, which is the thing that we would like to have, we are interested in. Then the formation of soluble complexes affected by chelators, formation of insoluble compounds affected by adding phosphorus. We are not going to touch upon this. They may be leached down to the water reservoirs beneath the soil level, and they can be held by inorganic soil particles, in which I would like to dedicate a few words. In clay, the particles in clay the cations are absorbed to negatively charged soil clay particles. You can see that the surface of the clay has got negative charges, so clearly it's the best place to be adhered to from that point of view. And so the task, the role of the plant roots will be to try to rob these particles, these cations, from the clay. How do they do it? We are going to see it immediately. And then there is another way, which is the fate of the soluble cations in the soil. It's being held by organic matter, affected by organic amendments. And this can be working on two manners regarding our interest. In the one hand, it can be chelating those minerals, those cations, in a way which will be very, very difficult to get, or sometimes it depends on the kind of organic matter, of the organic, not minerals, but the organic acids that they are there. And sometimes it's very easy to rob the organic matter, sometimes it's very difficult. Same holds true regarding the clay. So in order to take the iron out of the clay or out of the soil in general, we have already seen very many times that iron is very sensitive to the pH, to the environment, to the pH of the environment. And we can see here that the lower the pH of the soil solution, the higher the Fe availability to plant roots. And we can see here that ferrous iron, Fe2+2, is relatively soluble, but is readily oxidized to ferric iron, Fe3, which is insoluble in neutral and high pH that prevail in alkaline and in calcareous soils. Calcareous soils are soils that are rich in calcium, and they are also very basic pH-wise. Due to turning to Fe hydroxide, FeOH3, oxyhydroxide, and Fe oxides. The concentration of plant available ionic Fe2 in these soils is below 10 to the minus 15. So it's clear that it's barely taken up by the plant. Furthermore, in these soil types, iron readily reacts with phosphates, carbonates, calcium, magnesium, and hydroxide ions. So the products are highly insoluble. So even if we manage to release the iron for a moment, for a second, from the soil, there are so many enemies or so many competitors that will rob the iron again and won't let it be available to the plant. And here we can see in the middle, the lower the pH of the soil solution, the higher the Fe availability to plant roots. But we can see that iron, the same way, is also takes place also with other iron minerals, with iron nutrients, a similar trend like we see down lower part for other micronutrients like manganese, boron, copper, and zinc. However, there are also another trend, which is an opposite, and this holds true regarding phosphorus, potassium, sulfur, calcium, and molybdenum. So back to the 16 nutrients that Rachel just mentioned, just a little bit about how we categorize all these nutrients. Nitrogen, potassium, and phosphorus are nicknamed like macro because the sufficiency or the amount that is normally found in these in plants is between 15 and 40 milligram per gram, or 1.5 and 4% in the dry matter of the plant. Of course, the basis is always dry matter and not fresh matter, because water does not mean anything here. Then come the secondary nutrients, which are calcium, magnesium, sulfur, and chlorine, which is one order of magnitude less than the previous ones. And then come the micronutrients, whereby iron takes the most important share. But here we are talking about ppms, part per million. So no more percent, no more part per 1,000, but ppm, which is 1 to the power of 6. So critical leaf concentrations for sufficiency of mineral elements in normal crop plants, arranged by decreasing values. So this is again the entire table, the entire figure. Now, what happens if soil conditions prevent optimal Fe acquisition? Naturally, Fe deficiency will take place. And now I'm going again to the similar figures that Rachel just showed. This is, of course, I'm calling it plant leaves are talking to you, of course, by their own way, their own language. Of course, it's a hypothetical situation that all deficiencies show up concurrently, because such things don't take place. No plant can cope with such concurrent deficiencies altogether. So it's a kind of a synthetic way of showing the language of the plants. So nutrient deficiencies show distinct patterns on plant leaves, frequently as interveinal chlorosis. What do you mean by interveinal? We can see here the veins. Why again, I can't, okay, never mind. Everybody can see the veins on the leaves. It's clear to everybody what veins mean?
Yes.
All right. Unfortunately, I don't want to touch the the button, the red again, the mouse. But I hope it's clear that intervenous sclerosis show in different forms. For example, the ZN deficiency in the middle of the figure is very different from FE deficiency. And then they are also very different from the boron deficiency on the right-hand side up top. And now I'll make a claim. The incidence topography indicates nutrient mobility rate within the plant. Can anybody try to verify or contradict what I've just said?
Can you repeat the question again? I'm not sure we understood.
On the left, it says in green, the incidence topography indicates nutrient mobility rate within the plant. How does it coexist with what we see on the right-hand side? Okay. If we don't see it, for example, N deficiency down low there shows on the lower part of the twig, of the branch, because if there is an nitrogen deficiency, the plant takes steps and pass it on to the newest leaves. Okay, so the oldest leaves will be the first to show this symptom. However, FE deficiency will show up. Can anybody continue this way of this theme now?
It's higher and they're older?
Higher is younger. So this means that FE is barely mobile in the plant. So if it's the newest ones will not be able to get the Fe, the iron that has been accumulated in the older leaves. Is that clear?
Can I ask something?
Yeah.
Please.
You talked about nitrogen deficiency, and you said that the older leaves show the deficiency.
Right.
But if they don't have nitrogen, how do they grow more leaves? And
they suffer. They suffer. I don't want to go into details with all this, but such plants finally they die.
okay.
They cannot develop. They cannot make flowers. They're vegetative, and all other aspects of growth get lower and lower until it dies. But the most important thing that I would like to stress here, that this figure shows that Fe is barely mobile within the plant. Okay, because if it's all of this is concentrated in the lower leaves, it will barely go up and nurture the upper ones, which are the younger ones.
Now the question is, how do they go up?
There are two different manners. We'll look at them. One is the symplastic, and the other one is apoplastic. Symplastic is via the phloem. The apoplastic is via the xylem. We'll show it very soon.
Okay.
It will be slide 22. So this symptom specificity sends a clue to the grower. I mean, how these symptoms of the deficiencies sends a clue to the grower how to control the deficiency. If I see the ZN deficiency, which is very clear, or the N deficiency, or the S deficiency, which is very similar to the N deficiency, it's very easy to say, it's clear. This plant is suffering for that one, for that deficiency. However, but in case of simultaneous multiple nutrient deficiencies, the diagnosis might be much more complex. And I'm not going there. There are some ways to solve this problem and find out, like what we saw here. We can analyze what's the situation. We can chemically analyze what's the composition of the plant, and then we can see clearly what's the situation of calcium or magnesium, or before we said iron or zinc. So every one of them has its optimal level in the plant, and moreover, different plants have different preferences. So one should know exactly with what plant he deals, and then he will know exactly what's the optimum, what's the suboptimum, what's deficiency, or what might be the toxicity. So here we can see, again, plant leaves are talking to you. We can see the symptoms on cucumber, on strawberry, and on avocado. And we can see on the left one shows the deficiency. On the right-hand side, it shows the deficiency. So iron deficiency shows a distinct pattern of intraveinal chlorosis on plant leaves that is aggravated following deficiency intensity. Is it clear? I don't see you, so please, one volunteer at a time will say, okay, I can agree.
Yes, it is clear.
Thank you. However,
Oded, you better ask if anyone disagrees. And then if you don't hear anything,
no,
you know.
Shy. People are shy. So if one says, I agree, I understand, it's easier for people to express themselves, isn't it? Okay, so these symptoms are very nice. I mean, it's not nice, but sometimes it's only the part of the glacier that is shown up to us. But certainly there are much more difficult situation, like here. This is a 3D reconstruction of mitochondria from a cucumber plant. Up top, we see an Fe sufficient plant, and down we see an Fe deficient one. So iron deficiency damages essential cell compartments, such as mitochondria and chloroplasts. It thereby impairs the functionality of electron transport chains of the respiratory and photosynthetic apparatuses, which are Fe-dependent processes. The figure shows reduced thickness of the outer and inner mitochondrial membranes, as well as intense reduction in the number of the mitochondrial cristae, which are the folded structures that generate chemical energy in the form of ATP during cellular respiration. I think this figure talks for itself. So now we return to iron from soil to plant. So it's the root rhizosphere where nutrient uptake takes place by the plant. Here we go back to the clay particles that we saw before. We can see on the left-hand side the plant, and then the figure at the tip end. This is what it's blown up here. The clay particle, which is negatively charged, what we saw before. Then the mineral cations are released into the soil solution. How? Because the cations are exchanged in this situation, by hydrogen cations. And this is done by, as a rule, Fe is taken up preferentially compared with Fe3, but this also depends on the plant species. We'll look at it later on. And where do these hydrogen cations come from? They come from hydroxylic from carboxylic acids that the plant is excreting to the soil, exactly for this reason, for this purpose. So there are several agronomical methods for mitigation of Fe precipitation in calcareous soils, which, like we said before, are our enemies as growers. The first one is application of iron fertilizers by fertigation. For people who are not familiar with this term of fertigation, it's a combination of two words, fertilization and irrigation. So what we do is co-application with slightly acidic water solution and fertilizers via fertigation to locally reduce soil pH. So we inject a little bit of acid to the water and also apply the fertilizers into this water. So we place it's important to have placement of the fertigation solution in close vicinity to the root zone, thus reducing the probability that the Fe cations will encounter precipitation agents, like we said before, which will reduce its availability to the roots. So we apply the fertilizer solution frequently and in small amounts. So in ideal way, we can have nothing less than 24 sessions of fertigation a day, or sometimes even more. So if we just apply it just once a day, and the iron cations will probably meet our enemies, our competitors, which will not let it be accessible to the plant. Here we see how it's actually being done. On the left-hand side, we see fertilizer tanks, whereby we are producing dissolving the fertilizers into the tank, and then there are these blue pumps that pump a small part at a time of the concentrated solution which takes place, which is present in those tanks, go down the solution right into the main, the black pipe, which takes the water to the system, to the field. What happens? Now we see it here on the left-hand side. You see that irrigation is combined with a soluble fertilizer. So we can see that a diluted fertilizer is taking place all around the root area, the root volume of the plant. While, of course, on the right-hand side, we can see that somebody have broadcasted a granular fertilizer, which is, from our point of view, an inferior way, because if the water does not take parts of the granular fertilizers, there's a small chance that the plant roots will encounter those granular fertilizers, and finally the plant will not be able to absorb it. So no question that fertigation is a much superior way of taking part of the nutrition, the mineral nutrition of the plant. I hope this is clear as well. Any volunteer? Nobody? Do I have to repeat it?
No.
Yes.
Okay.
Want to repeat?
No. It's clear.
Okay. Another way of taking care of controlling Fe deficiency is application of iron fertilizers in the form of Fe chelates. For example, on the left-hand side, you can see an iron atom and an EDTA. Just in a second, I'll tell you what EDTA stands for. And then we can see that the iron is all of a sudden covered by this EDTA chelate, which protects it from being robbed by other factors within the soil, like we said before. So we can see here on the right-hand side a control plants that we can see that they suffer from chlorosis. This is the way we call plants that do not have the green, they don't have enough green pigment, chlorosis on the control. On the middle, we can see FE-EDTA, treated plants. And also there is another chelate, which is called FE-EDTA, which shows also a very nice development of the plants. So the plants here we can show here the effect of application of FE chelates on 21 days old cucumber plants. They're very, very small ones under hydroponic culture. So they're not planted on soil. We call it soilless or hydroponic culture. So we supply all the nutrients directly to the water solution in which the root system is developing. So this method is very useful also for foliar feeding. We can feed the plants exactly by the same way, not only via its root system, but also via its leaves. And by this, we can avoid the risk of the iron being taken up by our enemies, by competitors that are so ubiquitous within the soil. And here we can see different chelates that are in use. If anybody is interested in what it means, so EDTA is ethylene diamine tetraacetic acid. I'm not going to mention everything, but there are 5 major ones that are in use. EDTA is now the cheapest one to produce, and cheapest one that can be used. But it has some limitation. We can see on the table on the left that it is limited to the pH range of only 4 to 6, while IDA has very similar range, but on the other hand, it's fully biodegradable, which is an important factor right now, because we are trying to be to keep not to pollute the soil with things that might stay there for many years. So we are trying. It's now a very important faculty to be fully degradable. And then we have DTPA, which is active between 4 and 7. EDDHA that we mentioned before, it's very active in calcareous soils, and it can be used in open field fertigation or soil injection. And the best, or the most important, the most potent one is the HBED, which is active in the entire spectrum between pH of 4 and 12. So in really very difficult situation of calcareous soils, it has a long-lasting effect, and it can be applied to soilless application as well. It can be shown on the right.
And are all of these used today? All of these are used today in the industry to fixate the iron?
To control iron deficiency, yes.
But they're used today in the industry mass,
absolutely.
I guess.
In agriculture. If you call agriculture an industry, yeah, then you're right.
I see.
In the industry of growing plants, of growing crops, yeah. So we have got, finally, we have got a very nice array of agents that may be applied very successfully. Then
Odette, I want to add something, if you can show, apropos the question which I just asked you.
Yeah.
Not only for plant fortification, one is using the DTPA or the HBED. In the free form, they are used to remove excess iron from our body. So some of them have medical applications, not as the complex with iron, but the free DTPA or the free HBED. Moreover, DTPA is what is being used universally as a contrast agent together with some heavy metals. So when you get CT or some other type of X-ray forms, then you want to have a contrast or a cancer, for instance, they are using DTPA with some radioactive metal because it's a very strong bond. In our body, it's not released, and so this is being used normally in the medical field as a contrast agent because it's very stable. The plant is a different story. The plant can dislodge the iron from the DTPA, but in our case, we don't have similar systems. So we use it medically, just to give you the perspective about these compounds. Sorry, go ahead, Odette.
Perfectly. Perfecto. So there are some other methods by which we control Fe deficiency, mainly in the case of Fe precipitation in calcareous soils. On the left hand, we can see the way that Payon is a company, in a Belgian company that is very active in the field of phosphorus fertilizers. And we can see here a tomato greenhouse. On the left, February 13, we can see a crop, a plant that is very devoid of iron, so it shows sclerosis. Then on the center one, we see that the grower added micronutrient Fe. It is an NPK, fertilizer with a analysis of 0 N, 42% P, and 47% of potassium. This is the way we are calling the fertilizers. First N, then P, then K, and then it was also enriched with ferrum, with iron, 2.8%, and we can see that just 2 months later on, it became more dark green, and three months later, it has, after 3 months, it shows very nice dark color of the leaves. Now, how do the plant take up the iron? So plants have evolved 2 distinct mechanisms of obtaining Fe from the rhizosphere. Rhizosphere is the area surrounding the rhizo. Rhizo is, in Greek, it's the roots. To adapt to Fe deficient environments. The main difference between these strategies is the oxidation state of iron when taken up by the plant. Ferrous for strategy 1 and ferric for strategy 2. Strategy 1 works in dicots, dicotyledon plants. The knowledge about the components of this system is markedly more complete than that of strategy 2, due to the common usage of the dicot plant Arabidopsis thaliana as a model plant for the research of numerous molecular and physiological systems. We know that this plant has got a small number of chromosomes, and they have been all found out everything they know, people know everything about how what's the work up, work down, of the genomical makeup of these plants. So we know very much about it, and therefore the following slides I'll talk mainly on this strategy, while our knowledge is a little bit less regarding the strategy two plants. So strategy one, all dicots and non-graminaceous monocots respond to Fe deficiency in at least 3 of the following steps. Release to the rhizosphere of various metabolites, including organic acids that we mentioned before, which acidify the rhizosphere, and phenolics such as coumarins and flavins, which facilitate the reduction of ferric iron. In addition, secretion of the polyamine compound putrescine that appears to improve Fe plant cell penetration through its cell wall. 2, additionally, dicot plants produce and secrete chelators to the rhizosphere, which is actually a characteristic of strategy 2. And this is going to be a claim that we are going to make later, showing that in a way they are not so different, strategy 2 and strategy 1. Now, the third one is iron is liberated from the Fe3 chelates outside the cell by the action of a plasma membrane-bound surface ferric reduction oxidase. This is FRO, ferric reduction oxidase, reducing it into Fe2. Uptake of Fe2 by the high affinity metal transporter IRT1, iron regulated transporter 1, also localized on the plasma membrane and responsible for transporting the Fe2 into the root cells and thereby entering the plant's symplastic system. Here is what I referred to before. This is again on the left-hand side, we can see the Arabidopsis. On the left-hand side, it's a rhizosphere. On the right-hand side is the plant, the plant cell. So we can see that, first of all, on the upper part of this, we can see a protein, probably, this AHA2. I don't know if it's a protein or just the gene that enhances it, but anyway, the proton H goes out and tackles the Fe3 mineral, which is not soluble. So by this, it liberates Fe3 cations, and then together with the FRO2 that we just mentioned, it is reduced to Fe2, which is taken up by IRT1 and bringing it into the cell wall. In a parallel way, there is a scopolamine that produces BGLU42 that goes out again and uses also as a chelate. And therefore, this is also a way that iron gets into the cell. Now, on the right-hand side, we can see that there are different two main pathways by which the iron can come into the plant. Fe cations having entered the root via the apoplastic pathway, we can see it down in the red or in the violet way, whereby once it enters, or it doesn't enter the root hair, but it goes in between the cells. You see that on the right-hand side, it is the cortex of the plant, of the root. So it's a kind of לזחול באנגלית.
לזחול?
כן.
Crawl?
Crawl, okay, thank you. It may be described as crawling in between the cells. You can see it on the cortex with the red, signed with red. And finally, it has to pass into the cylinder, to the central cylinder of the root. This is the apoplastic way, the apoplastic pathway. While there is also a symplastic pathway by which the Fe enters into the root cell, the root hair, and goes within the plant cells, within the cortex cells, directly into the central cylinder of the root.
Oded,
yes.
Oded, there is a very important thing that I should mention.
Please.
Because you just jumped on it. The acidification of the medium that you get, it always occurs, or in most of the cases occurs, by a proton pump. So the AHA, as many others in bacteria, it's an actual acid secretion that you get by a proton pump. So this process is energy dependent because it's an ATP pump.
It's an ATPase.
Yeah, it's an ATPase.
A2, it's an ATPase.
Correct.
That inside breaks ATP and the proton are.
Yeah, but the important point is that you are really moving a proton is an active secretion.
Okay. So this is in general how
I have a question.
Please.
Does some symbiotic fungi doesn't play any role in the absorption of the iron?
Say it again, who doesn't have any role?
Any role, like symbiotic fungi? I had the impression, I don't know why, that fungi played a big role on this absorption, but I might be wrong. So.
The only thing that I may think of is that it's a kind of they fight for the. I don't think it has any negative effect on the plant itself, but they compete on this iron. So
it's more a competition than a help.
Yeah, it's there is no symbiosis on iron uptake. So that's in fact it's the opposite. There is a competition. The only case that there is a very tight symbiotic is on the nitrogen. Because the plant cannot, like iron, they cannot produce iron. The plant cannot fix nitrogen, and there is a symbiosis with rhizobium, with the bacteria. But on the iron, purely competition.
I see. Thank you.
So here we've got a kind of a very important slide. I would like you to remember it because we are going to rehearse and show it once again from another point of view. This is just, I'm showing you just, again, have a look at the left-hand side, down in the down. There's a cubicle showing a part of the root. So this is a soybean plant that suffers from Fe deficiency, which is shown in the very low part of the screen. And have a look down. There is a fuchsia square that shows what is going on within this section of the root. And we can see that this section of the root contains very, very many components that are very important regarding Fe, the uptake of the iron. For example, we can see on the within this blown up cubicle, you can see the TCA, the tricarboxylic cycle. Organic acids are leaving the plant, leaving the plant, going down into the soil, which is what we mentioned before, but by which it takes place by reducing the Fe3 into the Fe2. Here I can see also what you mentioned, Rachel, just now, the ATPase mechanism.
A proton pump. It's a proton pump.
Okay.
Not just an ATPase, it's a proton pump.
Okay, right. On the upper part on the left, we can see that all hormones that we know of are active in just this small part of the plant. We are talking just maybe every cell on the root have got all these apparatuses, which are the hormones like auxin, like cytokinin, like ethylene, like ABA, like salicylic acid, and on and on and on. So it's a very composite apparatus that also contain in the center of this cubicle the BHLH and the FIT and the FE genes that we are going to talk about a little bit later. But anyway, what the end product is the Fe2 that goes up from that part of the root up to the plant, and they enable the photosynthesis processes and growth. What I wanted to show you and to put in a very important stress, it's the FIT, the Fe deficiency induced transcription factor. The role of FIT in Fe homeostasis will be dealt with shortly. So this is a core slide. Now we are going to strategy 2. I'll try to make it more quickly because we are a little bit past of time. So Fe uptake by plant roots via strategy 2. Strategy 2, graminaceous monocots respond to the Fe deficiency in 4 steps. Biosynthesis of phytosidorophores, PS, that are Fe3 specific ligands of the mucineic acid, which is called by short MA, family within the roots. Secretion of these phytosidorophores into the rhizosphere, solubilization of insoluble Fe3 in soil by alkylation by the PSs, uptake by the roots of the ferric phytosidorophore complex, detachment of iron from its ligand occurs inside the cell by a yet unknown mechanism. But the differences between dicots and grasses is not completely dichotomic. As mentioned, dicot plants produce and secrete chelators to the rhizosphere, a characteristic of strategy 2. Peanut plants, which is a dicot plant, were found to absorb Fe3 PS complexes generated by PX secreted from neighboring maize plants. So it's a kind of a combination between a dicot and a monocot plant. And mucineic acid that we mentioned before, family PS, were detected also in olives, suggesting that non-graminaceous plants too can benefit from the PS system originally confined or theoretically confined to grasses. The co-expression network constructed from iron-responsive genes in rice transcriptomes reveals a remarkably similar architecture to the networks inferred for Arabidopsis, highlighting a high conformity of the iron deficiency response between the two species. And all these are mentioned by a relatively recent paper. So this is again a figure that actually shows what happened in strategy 2. We see it in a very similar way to what happened before in strategy 1, but here we can see that DMA that goes out and captures and chelates the Fe3, and then it comes back into the plant. So this is rice and barley were found to possess a functional homologue of IRT1, which mediates the uptake of Fe3 under low oxygen, thus blurring the distinction between strategy 1 and 2. Now, this shows something that happens. It is a combination, it's a synthesis of both apparatuses that we saw before. But it's important to know that soy plenum is occupied by a dense coexistence of plant roots and microorganisms, all competing for iron, zinc, manganese, phosphorus, etc. So once the complex Fe is in the soil, all organisms in the neighborhood may benefit from it, irrespective of who produced it. Therefore, orchards that are normally dicots can benefit from effects of DMA exudates of specific perennial grasses that are purposely planted as an intercrop. So we can see on the figure that very similar, in general, very similar apparatuses take place, but they actually compensate for each other. They compete and complement for each other, because, like I said before, soil plenum is full of different organisms, all producing and consuming the byproducts of each other. So here we can see an experiment. You can see that it was taken. It was pretty. The experiment was done as far away as long ago as 2010. I know this gentleman Rombola. It's in Italian, and he shows in this experiment on the left-hand side a control plant, then on the right-hand side an Fe chelated a medium, and in the center a Festuca rubra, which is a grass, which actually produces exactly the same product. Namely, it has green leaves, just as similarly to the Fe chelated plants. This is regarding how it looks like, the morphology or the phenotypic appearance. On the other hand, we can see on different plants total yield at harvest of blueberries under different treatments intended to control Fe deficiency. We can see the control on the left-hand side, rather low yield, Fe DDHA plant, treated plant, which is much higher, statistically different. Then Fe heme. Fe heme is a product of bovine blood compound, which is a kind of a fertilizer. It's an organic fertilizer. Then AB, very similar to Fe DDHA, is the same plant but enriched with Poa pratensis, which is another grass, or again the Festuca rubra, which is in this situation it gave the best yield. And I think
we stop here, okay?
Yeah. Okay, so Up till now, we've talked about how this iron gets into the soil, into the plant. Now we will talk up to the end of the presentation what it does, where it is located, and where does it go, and what it does within the plant. So Fe localization sites in the plant. Once inside the plant roots, Fe is complexed with organic acids such as citrate, malate, and others, and translocated to its specific activity sites in the plant cells, predominantly in the enzyme cofactors as components of the electron transport chains. Then nitrogen reduction and fixation and lignin formation, and loading into seeds and storage in the endosperms ferritin or in the aleuron vacuoles. We can see here the plant. We can see that it goes from the, it's being taken up by the roots. Then the red dots, we can see them on the leaves. These are Fe cations. The yellow dots are sulfide, and blue dots are oxygen. So we can see that it is the sinks of the iron would be mainly in the leaves, and some part of them also in the seeds, whereby it will be stored. In a minute, we can see it too. Fe citrate moves upwards from the roots to the shoot via the xylem with a transpiration stream. An important sink organ for iron is the leaves, where it is utilized for photosynthesis chelated enzymes. Iron is remobilized from the leaves and reaches other sink organs through the phloem, which is a part of the symplastic system. It is transported in the system in the form of Fe2, nicotinamine, Na complex. Na is a non-protein amino acid that is a precursor of deoxymogeneic acid that we mentioned before. Na and DMA are iron chelators, responsible for iron acquisition and translocation. Na functions in long distances as well as cellular iron transport in higher plants. An important Fe sink organ of the plant is its seeds, where available iron is indispensable during germination before the seedling has developed a root that would take up nutrients from the soil. Fe citrate malate complex has been identified as the principal iron form delivered to pea embryos.
Oded, there is something here that you need to explain to me, and because this is a general principle in biology. You said that iron is transported in the phloem as iron 2 nicotinamide. Now, iron 2 nicotinamide is a very, very weak complex. Is it certain it's iron 2 and not iron 3, at least with the oxymogeneic acid? Because my assumption will be that this is going to be very reactive iron. And usually iron is transported in a very safe manner. I just wonder.
Maybe I'm wrong. I can tell you later on when I go back to the paper itself, Chen et al. 2020. I will look at it and
because iron citrate for sure is iron 3.
Mm-hmm. Yeah,
that's the reason I wonder about the nicotinamide. Okay, go ahead. Sorry.
Okay, so here we can see how Fe iron is stored in the seeds. The way it is stored is in phytoferritin, which is the major form of iron 3 storage in seeds. It is reduced to Fe2 upon seed germination for initial energy production, prior to Fe acquisition by the first roots of the grain, of the seed. Here we can see a Prussian blue staining for iron in wheat grains from control line, top, and high iron line in the sperm in the bottom. Grains were dissected longitudinally, left, or transversely, right, using a platinum-coated blade. Will anybody tell me why I highlighted this fact, that the blade was platinum-coated?
Why not iron-coated?
Well, because we don't want any artifact with the iron, because the blade is made of iron. And if we cut it with iron, we will be misleading ourselves. Therefore, the part that is in contact with the grain, with the seed, will always be protected from being in contact with iron. Does it make sense? Hey, gentlemen, ladies. Okay.
Well, but maybe, Odette, I should maybe mention something very important. Everywhere in the biological field where they are going to see or want to detect any iron, you have to make sure that you are not contaminating with your own system. For instance, if you use glass has a lot of iron adsorbed to it. Definitely, if you are using an iron blade, many things have iron contamination. Therefore, in many cases, people have misled a particular property that is not present naturally, but was introduced by the method of detection. So it is very important that your detection method should be free of what you want to identify in your biological system. So this is a principle beyond this platinum-coated blade, I would say. Okay, but that's very general. You are going to see this.
Yeah. Before the slides that the previous slide was about storage. Now we are going to talk about the functional compounds and their roles in the plant. Fe2 is an electron donor and hence is a key constituent of electron transport chains. It is thus a component of cofactors that carry out electron transfer functions or facilitate chemical transitions such as hydroxylation, radical mediated rearrangement, and hydration or dehydration reactions. Iron cofactors also function in oxygen transport, oxygen or iron sensing, or regulation of protein stability. The most common forms of iron cofactors in plants are heme, FeS clusters, and d-iron centers. The toxic nature of free iron commands that the heme and FeS clusters biosynthesis is a highly controlled process. Compared to animals, plants contain relatively little heme, only about 0.5% to 1% of total iron. Heme contains only one iron atom inserted into an organic tetrapyral ring and is a non-covalently or covalently, in case of C-type cytochromes, bound to the protein. The most well-known heme proteins are the cytochromes, which contain a heme Fe porphyrin complex. We can see it on the right-hand side. Other heme proteins, peroxidases, are involved in the formation of lignin and suberin, and others in the breakdown of superoxide to water and oxygen to protect the cells from reactive oxygen species. Another way, the most common non-heme protein contains FeS clusters, which serve as cofactors for redox metabolic, catalytic and regulatory functions. They are concentrated mainly in the chloroplasts and in the mitochondria. The biosynthesis of FES clusters occurs in both the plastids and the mitochondria. Example of FES proteins are ferredoxin and FES-SOD. Ferredoxins are small proteins containing iron and sulfur atoms organized as iron-sulfur clusters. These biological capacitors can accept or discharge electrons with the effect of a change in the oxidation state of the iron atoms between +2 and +3. In this way, they act as electron transfer agents in biological redox reactions, such as in photosystem 1, which is the first stage of photosynthesis, reduction of nitrite by nitrite reductase, reduction of sulfite, sulfite SO3 -2 by sulfite reductase, and in biological and fixation. Here we can see a structural representation of an Fe2S2 ferredoxin. Another Fe functional compound and their role in the plant, we can see that we can have the superoxide O2-, which is a reactive oxygen species formed during normal aerobic metabolism, which is, if it's not eliminated, it disrupts cell enzymes, oxidizes lipids, and fragments DNA. And there is FeSOD, Fe superoxide dismutase, which is an antioxidant that converts superoxide into hydrogen peroxide and deoxygen. The reaction is called deproportionation. And we can see the reaction here down here, and a 3D image of an iron superoxide dismutase molecule, which is a complex protein. And the enzyme, of course, is a protein. Now we are going to have a little bit talk about the homeostasis in plants. Iron uptake, distribution, metabolism are controlled by tight regulatory mechanisms at the transcriptional and post-transcriptional level to avoid building of iron concentrations up to toxic levels. Iron homeostasis in plants is maintained through the action of 5 processes: high affinity uptake systems, transport and distribution, use in cofactors, which is the metabolism, storage mechanisms, and uptake blockage. Again, the sensitivity of different components of iron to the pH is very enhanced in plant systems. In plants, the Fe solubility varies in different cellular compartments. It is their pH that is the most influential factors. For example, in the different tissues, different pH and the solubility. In the xylem sap, which has a pH of 5.5, the solubility of Fe2 is immense. It is 80 mole per liter, while the solubility of Fe3 is rather small. It's about 3 and 10 to the power of -10. In the apoplast, which is just half the unit of pH, it has reduced tenfold solubility of Fe2. In the cytosol, which has a pH of 8, it has reduced 10 to the -4. So it's really very, very dramatic changes that makes whereby the pH is affected, the solubility, and therefore the activity and of the iron in the plant. Something more about the homeostasis. The role of the basic helix loop helix, BHLH, transcription factor, FIT, FIT, Fe deficiency induced transcription factor in Fe acquisition. You remember that we mentioned it in, I don't remember, it was slide 22 or 23. So FIT is not only essential, it is also a central regulatory hub in root cells to steer and adjust the rate of Fe uptake by the root in a changing environment. FIT regulates a subset of root Fe deficiency response genes. Based on the combination of co-expression network and FIT-dependent transcriptome analysis, we defined a set of FIT-dependent and FIT-independent gene expression signatures and co-expression clusters that encode specific functions in the Fe regulation and Fe homeostasis. These gene signatures serve as markers to integral novel regulatory factors and signals into the Fe deficiency response cascade. FIT forms a complex with the BHLH subgroup IB transcription factors. Furthermore, it interacts with key regulators from different signaling pathways that either activate or inhibit FIT function to adjust Fe acquisition to growth and environment constraints. Co-expression clusters and FIT protein interactions suggest a connection of Fe deficiency with ABA, abscisic acid responses, and root cell elongation processes. All this I cited from a recent paper by Schwartz and Bauer. And we can carry on seeing it again, the same figure that we saw before, and now the focus is on this FIT. And we see again that this is again talking about strategy one only, because it was found in the Arabidopsis, but we have already seen that there is some we have already seen that the distinction between strategy 2 and strategy 1 is not that clear-cut. So I put again this focus on this FIT that we saw before. So FIT is an Fe deficiency induced transcriptome factor. And here we can see on the next slide. Okay, first of all, let's talk about the FIT itself. It is a transcription factor. It is the master regulator of the iron deficiency responses in homeostasis in Arabidopsis. So I would say that this slide is one of the most important ones in the entire presentation, because we can see here that 15 different genes have been found to bind to the FIT in Arabidopsis. Their functions can be generally categorized into 3 groups, outlined in the next slide. They are A. The first group contains 4. I'm not going to say exactly what proteins, which bind to the FIT and act as positive regulators, stabilizing and activating the FIT in iron deficiency responses and homeostasis. B, the second group is so-and-so proteins that play as negative regulator by degradation of the FIT protein. C, the third group includes proteins which compete with the first group protein to bind the FIT and thus inactivate their functions, So this group has a negative effect too. And this is mentioned in the article that was published on 2 years ago. And I assume that the entire picture has developed furthermore. So to make it a little bit more, in a more plastic way, we can see a whole bunch of interreactions. And I'm not asking, and I don't think that anybody of us can detect who does what, but we can see that wherever such a situation takes place, that the brown is covering the fit, it means that it disguises it. It acts against the fit. While in this situation, where the fit covers this enzyme or this gene or this protein, it is the fit that can continue, that does its work, and it goes on and on all over here. And here, this is the system that, on the one hand, this disguises the action of the fit. On the other hand, the fit disguises this one. And altogether, we can see that the IRT is either functional or not. And all this system is found in the root tip. This is a steely cell. This is a steely part of the root tip. This is the epidermal cell. So all this composite complex apparatuses, all of them dwell within, I don't know if one cell or a group of cells, but something is very clear that this is the very center of the homeostasis regarding the activity of fit. Just to read it up a little bit again, once again, when root epidermal cells are under iron scarcity, various proteins serve as positive modulators, interacting with the key modulator fit in activation of the expression of iron acquisition genes such as so and so. Well, IRT is very important, FRO2 is very important as well. While under iron sufficiency, other proteins serve as negative modulators, competing with the first ones to bind FIT for prevention of excessive iron FIT. Of course, excess of iron has its downsides as well. And before we talked about what happens in the fit tip, in the root tip, sorry. Here is what happens in the steely cells. We can see something very different, very, very similar. Again, something that either...
Odette, before you take that, there is something here that needs some explanation.
Please.
And the explanation is, which is the sensor? Is fit the sensor for iron in the plant cell? If you don't have a sensor, I don't know what modulates what. So it's the iron level in the cell which is sensed by fit, or something else senses the iron in the cell in order to transmit or transduce the signal.
I think it's mentioned. I think it's mentioned later on. I think it also senses.
You mean FIT also is a sensor of iron?
Yeah, I think so.
That gets inactivated when it binds iron, I suppose. Because it's only in the absence of iron that it is active.
Absence or if it's there or if it's not there. It senses it, and the different or complex, there are complex reactions that either enhance it or subdue it.
Yes, but How is iron sensed in the cell is one of the questions.
I see the question. I don't remember exactly, but I think I covered it
Okay.
somewhere later on.
Okay.
So we see the same apparatus has been found in steely cells. Before we mentioned that it has been found in epidermal cells, but it has been found, same thing has been found also in steely cells. Okay, so this is something that maintains a homeostasis again from the point of view of the steely cells. And again, all this takes place, it has been found in the strategy 1 plants. We have to talk also about the role of phytoferritin regarding homeostasis. Ferritins are important iron storage proteins present across the biological kingdoms. About 80% of the plant iron is localized in the chloroplast in rapidly growing leaves. It is stored in the stroma of plastids as phytoferritin. It consists of a hollow, well-defined crystalline protein shell, which can store between 2,500 and 5,000 Fe3 cations, varying by species. Its concentration is high in dark-grown leaves, but it rapidly disappears during regreening and is very low in green leaves. As done before, can anybody support or contrast this claim? Phytoferritin is a storage system. So it is high in dark-grown leaves, but it is very low in green leaves because then they are using it as a cofactor for their enzymes. Is this clear? I hope it is. Okay. In young leaf tissues, ferritin-bound Fe features an important Fe source for biosynthesis of Fe-containing proteins in photosynthesis. Phytoferritin is a vital compound in maintenance of Fe homeostasis and in protection against oxidative damage catalyzed by Fe-induced formation of ROS that are byproducts of a normal aerobic metabolism. Example given, peroxides, superoxides, hydroxyl radicals, singlet, oxygen. Phytoferritin is present also in the xylem and phloem and is abundant in seeds. See slide 33, we've seen it before. Now there is a whole bunch of regulation genes and expression pattern, and I'm not going to go into details because for all this. And I must mention that there is less information about the transcriptional networks governing iron uptake in monocotyledons plants, which is the strategy 2. So we will skip this, but on the other hand, we can see that there are some genes that we are familiar with already. So a regulation of iron homeostasis by morphological means. Before we talked about genetic transcription and post-transcriptional methods. Now morphological means. Ensuring that sufficient iron reaches the tissues where it is needed without accumulating to toxic levels commands the existence of an efficient regulatory system that senses the iron status of the plant and adjusts homeostasis accordingly. Plants adapt their root morphology to iron-limiting conditions by increasing the density of root hairs and the number of lateral roots. The greater surface area extends contact between the root hairs, epidermis, and the rhizosphere, and the lateral roots help to explore uninhabited soil. But this development might challenge the homeostasis, leading to Fe overload if iron becomes suddenly available excessively in the environment, for example, after rainfall, when a large amount of iron becomes soluble. The entry of iron into the symplast is controlled at the endodermis and the thickness of the sugarized cell walls, and we have seen it in slide 25. Remember the endodermis and the pericycle and the central cylinder. And several post-transcriptional mechanisms have been observed that rapidly stop iron uptake, like the IRT1 is continuously recycled from the plasma membrane, in addition to swiftly degrading the uptake machinery. Stopping the transcriptional response has been found. A small family of hemiferritin E3 ligases, including some Brutus BTS, which I don't know, in Arabidopsis, and HRZ in rice, which is a monocote, may sense iron and act as negative regulators of the iron deficiency response. However, there are still a number of questions to be resolved regarding the homeostasis. The precise molecular mechanisms that link iron availability.
Odette, before you move this, can you go up again to what you just showed? Now, this is the most important slide, I think, from the point of view of homeostasis, because what you are saying there in 4, okay, is that there is a small group, a family of emeritrin E3 ligases. We are going to see the same thing in mammalian cells. Those are hemoglobin-like. It's a very primitive hemoglobin-like molecule. It's not really with the heme, but it's emeritrin. I showed that in the last lecture. And those can be sensors of the iron in the system. In fact, it's iron and oxygen, and they can be the ones that will transmit it, to the genes themselves. Namely, they are, iron responsive elements in different genes, and this is, the important thing that connects between a plant and a mammalian cell. They sense the level of the iron according to the oxidation stage that you have in the medium that is caused by the presence of iron. They transmit this information. In this case, it will be, to the fit, which is what you mentioned, before, and those will be negative regulators in the presence of iron, and of course, positive regulators in its absence. So this is exactly what I was driving. And it's very important because I want to see a unified thesis, what you have in bacteria, in plants, as well as in humans.
Mm-hmm. I admit I didn't know all about this, and I'm happy that I found something that could connect the systems, the higher animals, higher organisms with the relatively low ones. So I admit that's my ignorance, that I skipped it without.
This is the point. We want just to add, everyone adds from its own knowledge a little bit.
Okay, good. So again, the questions that have to be resolved. The precise molecular mechanisms that link iron availability to changes in, okay, this is something that I mentioned already. No, so I'll repeat it. The precise molecular mechanisms that link iron availability to changes in root morphology or permeability are not yet known. The substrate specificities of many transporters involved in iron transport, cell to cell and between intracellular compartments, need to be demonstrated by biochemical studies. Unknown components of long-distance signaling between shoots and roots, specifically how the iron status is signaled to permeability changes of the endodermis. And many redundancies between gene functions have been reported, and a unified model of the signaling cascade is lacking for the time being. Now, I wanted to touch upon one issue that Rachel has mentioned, which is the agronomical way of controlling iron deficiency. Biofortification of edible crops. Biofortification of staple food crops is one of the most efficient strategies for providing sufficient level of iron and other important dietary components to vulnerable populations. Cassava or tapioca or manioc ranks 5th among crops directly consumed by humans. In Africa, cassava is a central source of starch for people, but it is very low in micronutrients, placing millions who rely on this crop at malnutrition risk. These photos show a plantation of, which is a small shrub that has these kind of roots, very, very fat roots, and like in potatoes or batatas, here it's transversion, they're cut, and they contain a lot of starch. So it can be used when it's cooked or even raw. And I was there in this symposium when this gentleman, Narayanan, from the United States, presented his findings that he could do a transgenic modification to the plant, whereby it increased the Fe contents in cassava field roots at 3 to 4-fold in dry matter, concurrently with 3 to 6-fold higher Zn concentration, without altering plants' productivity. And these are achievements very, very important to people who live in such difficult situations where they are deprived of many important micronutrients, both for the plants and for the people consuming them. Now we'll be finishing with 4 slides regarding toxicity. Situations generating iron toxicity in plants. Acid sulfate soils are rich in iron sulfide, found as microcrystalline minerals such as pyrite. They're formed naturally in soils where long-term waterlogged conditions occur, such as estuaries, wetlands, and shallow groundwater in deep sands. They feature pH lower than 5 and contain massive amounts of Fe at 2,000 milligram per kilogram. Fe ions are highly soluble in water. FeOH2, the KSP of this is 7.9, 10 to the minus 15 at 25 degrees. Hence, all Fe2 ions that exist in low pH soils are soluble in the soil solution available to plants. Main soil types prone to iron toxicity are acid sulfate soil, acid clay soil, and peat soils. Acid soils occupy approximately 4 billion hectares, featuring 30% of the total landmass in the world, representing more than 50% of the potential arable lands. So it's really a real problem from an agricultural point of view. Thus, Fe toxicity is one of the major limiting factors of crop yields and quality, particularly in China, India, Southeast Asia, West and Central Africa, and Brazil. The sequential effects of Fe toxicity in plants. In submerged soils with anaerobic and low pH conditions, ferric ion is reduced to a remarkably more soluble state of ferrous ion. Excess ferrous ion is absorbed by the roots, where it inhibits cell division and elongation of the primary roots, and subsequently it inhibits the growth of lateral roots. Fe2 is also transported by the xylem to the leaves, causing Fe overload in plant tissues. Fe overload in the leaves impairs photosystem 2 by tackling cytochrome functioning and accelerating oxygen production. In the chloroplasts, it causes overproduction of ROS by the Fenton reaction. These ROS damage cellular structures, membranes, DNA, and proteins, and impair physiological processes. The outcome of all the above create bronzing symptoms in leaves, followed by loss of yield and quality. Here's again, plant leaves are still talking to us, but here on the other direction, iron toxicity shows a distinct pattern of leaf bronzing. Excessive Fe is deleterious because free Fe2 reacts with reactive oxygen species produced under normal respiration and in response to stress factors. The symptoms are shown on plant leaves are aggravated following toxicity intensity. Here you can see in the soybean, more than anything else, how it becomes bronzing, and also the chlorotic symptoms that show that actually these leaves cannot function. Oops, why did I do that? Okay. I'll try to cut on my continuing. There's a special situation regarding rice. Lowland rice is typically grown in submerged paddy fields with low soil pH, where highly soluble ferrous iron is abundant. We can see here that the large concentrations of Fe in submerged acidic soils. They produce an Fe overload in the plant tissues, overproduction of ROS, physiological disorders like damages in cells, DNA and proteins, bronzing symptoms on leaves, and yield loss or crop failure. But rice has got its own means to protect itself. Rice employs 4 defense systems to maintain Fe homeostasis under these conditions. They are Fe exclusion by suppression of genes involved in Fe uptake and translocation. It is the Fe binding ubiquitin ligase, HRZ, that serves as a key regulator that represses Fe uptake genes. 2, retaining Fe in the root system rather than transporting it to the shoots. 3, compartmentalization of the Fe in the shoot. In defense system 2 or 3, 3 factors mediate the isolation of detoxification of excess Fe. They are vacuolar Fe transporter, so-and-so, Fe storage protein ferritin, and the nicotinamine synthase, and the detoxification of the ROS produced within the plant tissues. And we can see it in a more graphical way. There is the defense system 1, whereby Fe exclusion takes place by the roots. So although there is a lot of Fe in the neighborhood, the roots are blocking their uptake. Then the defense 2, Fe retention in the roots. They fill up with this Fe, but they don't let it go up into the shoots. Then there is the Fe mitigation within the shoots. Everything goes with all its apparatuses. And then there is defense 4, whereby ROS is detoxified. And I think that by this, we finished our presentation. Sorry. Are you still there?
Yeah. You went to bring something to drink, probably. Yoav, אתה שם?
Odette, can you show the before the last slide? You went over the most important slide. No, the next.
That's one?
Yeah, because here you are actually summarizing pretty much a whole strategy. I want to use this because this is where we are going to hear a lot about in the next lectures. So what you are showing is an important factor, that plants have no ways to get rid of excess iron. That's a very important thing. But the only thing they can do, they can control how much iron they take up, even when they have an excess of iron in the medium. So they know how to control their levels. And number 2, if for some reason you get too much iron into the plant, they compartmentalize it so as not to bring any damage. This is second defense that you have. And the third defense, or fourth, if you like, if there is any damage caused by oxidative stress, to correct the damage by different mechanisms, let's say ROS detoxification. In other words, this is a biological strategy of different defense forms that you have, okay? Either preventing it from flowing from the roots, not taking up at all, compartmentalizing it, and in the case there is a damage, to control the damage. Because that's exactly what we have in all the animal systems. We cannot control, okay, excess by removing excess iron, but we can deal with it by trying to prevent it or trying to compartmentalize it, so in order for that not to be toxic. So those are 2 or 3 important defense mechanisms that we all use universally. So this would be a good point to stop, I would say, because there is a unified hypothesis in terms of dealing with iron. We are using similar strategies. Each of the strategies has to be adapted to the proper condition where the iron is present. And we have all the internal mechanisms inside the cells or in the entire organism to distribute it within the cell or compartmentalize it, depending on the needs of the cell. So this is an important thing, that I would like to stress as the end point of your presentation, because this is going to connect to the next one that we are going to see from next week onwards, when we are going to deal with animal cells, homeostasis, cells, and later, of course, in organisms and the kind of diseases that we have. So any questions from the students, or are they already overridden by plant iron? Well, it looks like you are overridden by plant iron. So at this point, I would like to thank Odette for this tour de force that it took us in plants. We don't expect you to remember so many different details, but maybe of the important things that some of them he repeated because we have already seen them in introductory lectures, but now a little bit more expanded. You got some sort of a flavor for what homeostasis is, although we have not reached in plants the level of sophistication as we have reached in bacteria, for instance, where we have a full understanding of the homeostasis in bacteria. But we have learned about new mechanisms in plants, and this is very important because plants, being non-mobile, have their own way to deal with the acquisition of the metal from the medium, according to the properties of the medium, and of course within the plant itself with all the complexities that you have heard today. So I would like to thank again Rachel that presented, introduced the field, and the tour that Odette took us in the plant world as far as ion is concerned.
Yeah, I want to say two minor things. One, plant cannot be compared to bacteria because bacteria are one cell organisms and plants are a whole organism, like, you know, like animal. It's an organism, and the bacteria are a unicellular organism, and to control a whole organism is exactly the same problem that we have in mammalians. The other thing that I want to say is something that is emerging in the field of the plants. There are a lot of efforts invested because they think that